Greetings from the 85th Cold Spring Harbor Symposium on Quantitative Biology. This year, the topic is biological timekeeping, and the meeting is being held virtually for the first time. That's why you see my house instead of the beautiful Cold Spring Harbor behind me. I'm Brian Ray. I'm a senior editor at Science Magazine, published by the American Association for the Advancement of Science. And I've had the pleasure of attending this very distinguished meeting and learning about biological timekeeping. And I'm very pleased to have here with me Luis de Lecea, who is a professor in the Department of Psychiatry and Behavioral Sciences at Stanford. And what we're going to talk about is mainly sleep. Of course, one of the most important things about biological timing being that we have to put aside some time for this kind of mysterious process of sleep, which I think we really don't understand real well, except we know that there are these regenerative properties that are really necessary for proper function, and that problems with sleep cause a lot of medical issues as well. So actually about 20 years ago, Luis was one of the discoverers of a neuropeptide that's called hypocretin or orexin. And neuropeptides are a kind of neurotransmitter that are produced by cells in the brain, and they can communicate to the other cells around them. And this set of neurons that makes hypocretin in the brain is quite a remarkable set of neurons. They have these effects on sleep and arousal that we'll be talking about, and they also control things like feeding and even emotional state and reward pathways. So really quite an interesting set of neurons. But in terms of sleep, the malfunction of these neurons is associated with narcolepsy, this problem where people have excessive amounts of sleep and actually sort of fall asleep doing things. And so we all hear about this sort of importance of getting enough sleep. But Luis told us at the meeting that these hypocretin excreting neurons may actually have a role in one of the big problems that we see in people during aging. And that is that the brain function of older people seems to deteriorate, and they have trouble with memory processes as well. And so I'm hoping that Luis can give us a little insight into how he connects these hypocretin secreting neurons to this process of problems with memory and aging.
Sure, my pleasure, Brian. It was indeed a very interesting meeting, and I was honored to be part of the sleep panel, where we described our most recent data, where that implicate indeed this group of neurons, the hypocretin orexin neurons, in sleep continuity associated with aging. So we all know that the sleep quality during aging is compromised, and one of the hypotheses is that the reason why cognitive sleep fragmentation accompanies cognitive decline, and the hypothesis that the reason why sleep fragmentation accompanies cognitive decline is because one needs a certain amount of sleep in order to consolidate memories. And when sleep is disrupted, naturally or artificially, then there are consequences on the information that the brain processes, and possibly accelerates the cognitive decline. So we hypothesize that one of the mechanisms by which that could underlie the sleep fragmentation in aging was the fact that the neural circuits that modulate excuse me, that modulate sleep and wakefulness are impaired or malfunctioning in aged individuals. So we considered several hypotheses. We considered that that the brain is just degenerating non-specifically, and one of the neurons one of the systems affected by this neurodegeneration happens to be involved in sleep and wakefulness. But we found that it's that this effect is that the neurodegenerative effect is not non-selective, so it really affects particular groups of neurons. And we were surprised to see that one of the really sensitive groups of neurons is the hypocretin ensemble in the hypothalamus. Indeed, we saw that in aged animals, we saw a 20% decrease in the number of hypocretin neurons. But the remaining cells we also observed that the cells that were remaining were functioning in a different way than normal neurons, in the sense that they were more excitable. They were closer to action potential threshold, as we call it. So they were easier to activate, we conclude that because these neurons are so hypersensitive, that it's just easier for the brain to wake up in many different conditions, and this is one of the reasons why sleep is disrupted.
Yeah, so that's really interesting. So it seems like these particular neurons in the brain could be extra active and producing this arousal state when they shouldn't be. And you told us that you've gone even further to look at a particular ion channel in the membrane of these cells that participates in the regulation of the excitability of a neuron, and that ion channel might be responsible for this hyperexcited state, and even that you can do some manipulations to that channel that would return that state of that cell to something more like the young animal. Can you tell us a little more about that?
Yeah, so that was all work done by my postdoctoral fellow in my lab, Shibin Lee, who had the really wonderful idea of looking at many different, potential culprits of this hyperexcitability, and he landed on this family of potassium channels, voltage-dependent potassium channels, that essentially are responsible for the resting memory potential. And without getting into too many details, these channels are different from others in the sense that they don't affect the firing properties per se. So neurons can still be active and fire normally, only that they're, I guess, as I mentioned earlier, closer to threshold. So they're just easier to activate. And there are examples in the literature where these voltage-dependent potassium channels, known as the KCNQ channels, are all affected by aging. For instance, in C. elegans, in this worm, people have seen that these KCNQ channels dimerize and oxidize and so dimerize normally, and during aging they get oxidized, and their function is impaired. So we haven't been able to prove that in mice yet, but there seems to be a sort of natural aging process for these channels, and they get they just their conductance is just reduced. So yeah, as you mentioned, we had then the idea of to manipulate these channels pharmacologically. There are actually a few molecules, small molecules that are known to activate those channels specifically. So we decided to see whether these molecules would have an effect on the excitability of the hyperactive neurons in particular, and more generally on sleep architecture in aged animals. And indeed, we were able to rejuvenate, as you mentioned, the sleep patterns of aged animals using this simple compound. These pharmacological agents have been used actually in the clinic for a while to treat epilepsy. So they reduce the intrinsic excitability of essentially all neurons. But they don't, as I mentioned earlier, they don't affect the firing properties. So it's not that the normal, firing patterns are going to be affected. It's just excitability is slightly modified, and of course that in epilepsy is quite important. These molecules, these small molecules, have a relatively long track record in the clinic for the treatment of epilepsy. So we took advantage of that and administered the compound to mice and saw a very, very nice reduction in sleep fragmentation in the aged animals.
That's impressive.
Yeah, thanks. So yeah, it's also interesting that because these anti-epileptics that are based on KCNQ channel opening have been in the clinic for a while, we were curious to see how effective they were and whether they had any side effects or interestingly enough, the biggest complaint for these compounds was somnolence. So, of course, we don't have clinical trials that directly prove that these compounds would be effective at treating sleep fragmentation in aged humans. But there are some indications that they are actually working quite well in that in that regard.
All right. Well, thank you very much. I mean, it's a really spectacular example of sort of getting at the molecular basis by which these sorts of timing mechanisms are working, and how the control of these timing mechanisms is having a real impact on physiology and behavior and actually health problems, and also this process of aging. So thank you for taking the time to explain that to us.
Thank you.